So hello and welcome to another episode of Interviews with Experts. Today, my very special guest, Dr. Margarita Lopez-Uribe, is an entomologist who serves as the Lorenzo Langstroth Early Career Associate Professor of Entomology at Pennsylvania State University. She also works as an extension specialist in pollinator health. Her research and extension work focus on understanding how agricultural practices impact the health and evolution of bees, with the goal of developing more sustainable management and breeding programs for both crops and pollinators. Her research uses molecular ecology to study how human-driven environmental changes affect both wild and managed bees. She has worked with different bee species, including honeybees and squash bees. She's also interested in using citizen science to engage the public in pollinator research. Dr. Lopez-Uribe holds a Bachelor of Science degree in biology from the Universidad de los Andes in Colombia, a Master of Science in Genetics and Evolution from the Universidad Federal de São Carlos in Brazil, and a PhD in entomology from Cornell University. Before her current position, she was an NSF postdoctoral fellow at North Carolina State University. I hope you'll stick around for this very interesting conversation. I'm Frederick Dunn, and this is The Way to Bee. First of all, I want to thank you for joining us and being here on The Way to Bee Interviews with Experts. And we just want to remind people that this is also a podcast. So if you're driving somewhere and you're watching this, please don't wreck your car. Just Google The Way to Bee podcast and you will find today's episode. So I want to thank you for being here, and I appreciate it because I have wanted this interview for a very long time. And so if you would, please just take a moment and introduce yourself and tell us where you are, what you do.
Hi, Fred. Yeah, so my name is Margarita Lopez-Uribe. I'm an associate professor at Penn State University, Department of Entomology, and I am also the extension specialist on pollinator health here at Penn State.
And so that was brief. So thanks a lot. And one of the things I really want to talk to you about is one of the studies that I don't know if it's concluded or if it's ongoing, but this involves feral colonies of honeybees that are existing in different tree cavities. Is it around the state of Pennsylvania, or just please describe kind of the scope of that study?
Yeah, so well, the study that you're mentioning focused on, like colonies we found across Pennsylvania, and it involved community scientists in the project. So yeah, but the root, or like the origin of this study was actually it started when I was a postdoc at NC State in the lab of Dr. David Tarpey. And so I was involved in a project that was actually comparing parasite levels and immune responses in feral and managed colonies in urban settings. And they found that overall, they didn't really see huge differences in terms of parasites, but they captured something similar that we also found in this other study in Pennsylvania regarding differences in the immune gene expression of feral and managed colonies. So basically that led me to become really, really interested in feral bees, because as an evolutionary biologist, right, like thinking of feral colonies had, like a very, like spark some interesting questions, particularly because, like colonies that live in feral environments have, recent connection to, management and domestication. So I was really puzzled by this idea, right? Like, how is it that these colonies that escape these management settings are dealing with pathogens? When we manage colonies in apiaries, beekeepers are actively there's a lot of variation in this, but generally speaking, beekeepers are actively managing parasites in, pathogens. And when these colonies escape these settings, right, and establish in the wild, they don't have beekeepers, like helping them with, like these natural pressures. So we were wondering, what are the bees doing? And so that led to this study. But one of the challenges of studying feral bees is that it is very difficult to see them and find them. So this is where we heavily relied on beekeepers across the state. So we did a lot of outreach, and dissemination when we gave talks at, clubs or the state meeting. And we just had a great response. We, had dozens of reports for the 3 years that we were collecting data, and were able to follow about 30 colonies over the course of the study. Feral colonies, many of them died, many of them were reported, and we would go and visit the the trees or the location where they were reported, and in the spring we would find empty cavities. But the ones that survived, were able to sample them over time and analyze levels of viruses and immune gene expression to compare them.
So what was the time frame? When did this actually start? After people reported the colonies that they knew about, are we talking specifically in trees, or were they also feral in abandoned buildings and things like that?
Yeah, everything was included. So yeah, there was a great variation in the cavities that they were that we found them in. And so let me think about this. So we started the project in 2017. So we started the outreach and communication for the project started in 2016. And we received the first reports that winter. So we checked some of the colonies in 2017 in the spring, and the ones that were alive, then we recorded them as feral because they had, survived at least one winter in the wild. And so the same colonies were sampled in the spring, in the fall of 2017. And then some of them survived that second winter and were sampled 2018, spring and fall. And we had new reports. So actually the sample sizes for the second year are a little bit, larger.
So what was the number that survived percentage-wise? If we're talking, you said you had some that
Yeah.
went 2 winters out of 30 initial colonies. How many made it through the second winter?
Yeah. I think that the survival rates were similar for both. So that was one of the interesting findings of the study. That, our prediction was that because of the lack of pest control and management in the colonies, we were going to see greater losses in feral colonies. But what we found was actually very comparable. And I think I want to say that it was somewhere between 40 and 50% for both years. There were some differences, but it was pretty high for both years.
Did we get any that surprised you and went 3, 4, or 5 years?
We did not follow colonies beyond, the second year for logistical reasons. But yeah, I mean, I think it would be extremely interesting to do that. And in some ways, because of this project, we connected with beekeepers who have been keeping colonies alive without, treatment for breeding purposes. And we know of a few beekeepers who, have shared stories of colonies surviving, multiple years without any treatment. Usually, there are certain characteristics, like they're quite isolated and the colonies are kept very small, right? So I think one of the key things to keep in mind when we're talking about keeping colonies without treatment is that oftentimes those those colonies are used for breeding purposes and not as, like honey producing
Mhm.
operations. And I think there are big differences in, like what you can achieve for one or the other.
Mhm. So you're saying that there people that are doing treatment-free management are selecting from their overall stock, finding the most successful colonies, using those queens as their breeder queens, not necessarily using these colonies for production or honey or pollination services. It's mostly focused on providing queens and genetics.
I mean, the couple of beekeepers who we have worked with for a couple of other projects, yes, that would be um the right, like description of those operations. And indeed, we actually at a very recent, like similar time frame, we started another project on organic beekeeping. And
Okay.
we actually compared the outputs of, like organic versus conventional versus treatment free for honey producing operations. And yeah, and the results were pretty, like convinced us very, like clearly that it is very difficult to keep honey producing colonies without any mite treatments.
Without my treatments. And
Mhm.
it seemed like, didn't the organic, or I think that study was reported on by Dr. Underwood, is that right?
Yeah, yeah,
And so the treatment free, the organic treatments, so management of mites through organics like oxalic acid, formic, and things like that. And then, of course, the probably synthesized chemicals that were used for treatment. And the organics outperformed the other two, is that right?
well, they didn't really outperform in terms of survival of the colonies. It was very comparable, right? And so I think what we learn is that for I guess I just want to be mindful with, like the scale of operations and the purpose of operations, because I don't want to, give generalizable statements. But for kind of like mid-size operations, honey producing, non-migratory, you don't really need to use synthetic miticides to keep your colonies, like mites at check and, healthy, uh, like productive, like colonies. So that was the big take home message for that study. But then we just published another study a few months ago where we actually did an economic analysis of, those same colonies of the same experiment. And we actually see a small um, but significant difference in honey production. So indeed, the organic colonies in that experiment produce more honey than the conventional colonies. And so when we did projections over time, what that meant is that the organic management system was more profitable than the conventional one. So there are, we do see some differences. Again, these are, like smaller operations, non-migratory, but it's very exciting because it indicates that there are some options for, like smaller beekeepers out there to produce honey without any synthetic miticides in their products.
Hmm. And so going back to the bees and trees, um, I tried to track some of them here. I live in Erie County, northwestern PA, and we had 10 colonies in trees. And I thought I would be excited just do my own, take a look at them with an endoscope and see what's going on. And hopefully they would be around. But I had similar results to yours, except that by the second year they were all gone. Like the trees that I was looking into, they were just empty and didn't repopulate.
Mm.
The other thing is often they're repopulating in spring from, managed colonies nearby that are going into these trees again and starting over. So one of my questions, I guess, is was there any way to identify the queens that were in those colonies that we were monitoring in trees to know that those are the same trees, or maybe they had been superseded, or they had swarmed and been replaced? How would we know that that might have occurred or not?
Yeah, so at the time of when the study was published, we did not have that information, but we did follow up we did, and this is not published yet, but we sequenced the genomes of workers of these colonies in collaboration with Brock Harpur from Purdue University. And we found some, like interesting patterns, like in some areas of Pennsylvania. The details are a little bit blurry right now, but something interesting we found where there were some indication of higher proportions of A lineage parts of the genome in the feral colonies compared to managed colonies. So that, like there could be some interesting processes undergoing in these, like feral colonies. But going back to your question about the identity of the queens, one of the things that we learned is that we included two colonies that in this paper are described as colonies that survived both years. But with the genetics, we actually found out that the queens were different. So at some point, throughout those 2 years, the genetics of that colony actually, like were replaced, or, probably the first colony died and, like a new colony moved in. It's unclear. But yeah, so I think what you're describing is probably something very common. 1 year survival, happens often, but I think, rarely the colonies really make it, over multiple years.
Mm-hmm. So what was the final kind of takeaway from the study? The study's concluded, is that right? So it's not ongoing?
Yeah, I mean, this study again, like led us to like other lines of, like research inquiry, but we are not currently studying feral colonies anymore. But I think it was, like something that was very clear from the data. And I was actually just, like reading a recent paper going a little bit, like farther with, like what I'm about to share, is that oftentimes these feral colonies evolve mechanisms of tolerance, right? And so what they do is that basically they can cope with the presence of high levels of viruses while reducing, like the fitness consequences of these infections. And so that, like leads to maybe colonies being able to survive, 1 or 2 winters, right? But the problem is that then you can think about these colonies as, machines that are just, like helping these viruses, like replicate at crazy levels, right? And so, and this is one of the problems that we have right now with, like mites and viruses, right? Like the mites have helped deforming virus and associated viruses. Just, these are machines of replication. And we have so many of these viruses in the environment that, we find them in other insects, right? Like not only other bees, but roaches, wasps, beetles, right? Like anything around, like honeybee colonies is probably, like exposed to these viruses. So yeah, it's an ecological bigger.
But that's kind of, it's interesting that you say that, because so with the fact that honeybees have such a potential for rapid reproduction and therefore gene selection, and the next generation just keeps coming out. Likewise, these viruses are living organisms that are adapting and changing, right? So they're kind of, are they outpacing the bees?
Well, I mean, they are definitely evolving more rapidly than the bees, right? Like we have new strains, more virulent strains, right? And so keep in mind that for, I don't know exactly what the number is, but for every like bee generation, there are probably dozens of generations of the viruses, right, occurring. So yeah, I think the replication rate is, like significantly higher, which means that they have more opportunities to change and adapt than the bees do.
That is not good news.
So, it's not.
Because that my audience is largely backyard or sideline beekeepers, And of course, they're all chasing, the idea that we could someday have genetics or something where we wouldn't have to treat the bees, but integrated pest management as far as physical management equipment and screen bottom boards and things like that
Mhm.
would someday be enough. What's your, kind of off the shelf opinion about, can you be a backyard, small scale, treatment free beekeeper and hope for some success? As far as, maybe you just don't ever get honey, but you're going to have so many swarm cycles that they somehow magically survive every year. So these smaller colonies, but kind of based on what you're saying, is eventually these virus loads will catch up because they're progressing faster than the bees themselves can genetically adapt and resist, so they become tolerant. And so we have these mite tolerant bees also that are survivor stock, but would it eventually, they would end up dying out? I mean,
Um,
can we go to the one surviving colony that's on its own and have a hope that that will ultimately seed the remaining genetics that would recover somehow? They talk about the Arnott Forest that Tom Seeley
hmm.
discusses, this survivor line or Darwinian, let them die and let those that live live, but you could eventually end up with no bee stock.
Yeah, and and I think from I stopped reading about the art of forest bees a few years ago, but like up to maybe like 2022, um, my impression from the work that Tom Seeley published was that colony size seemed to be a very important trait of those,
Mhm.
right? Like of those colonies in the Arctic forest, right? Like, so one of the big findings was that the tree cavities were really small.
Mhm.
And so, like just the capacity of those tree cavities was, like they couldn't really let the colony just go, crazy large, like what beekeepers can keep, just stacking more boxes, right?
Right.
Um, and so, in that sense, that like again, all of this is important for beekeepers to consider the context of how is it that you can keep Darwinian like beekeeping going. Are you going to be reducing the size of your colony? Are you okay with losing 50% or more of your colonies every year because the losses are high, right? And that's something that, I mean, going back to our organic study, we we started the project with 96 colonies in each treatment. So the treatment free treatment had 96 colonies on year one. I think we had I don't remember the exact number, but we had something like 5 or 3 colonies that were able to survive without any treatment the 3 years, right? So it can happen, right? It does happen. But if you're a beekeeper who, is trying to, get some honey out of the colonies or not spend, like hundreds and hundreds of dollars every year replacing your colonies, is that something that you can afford to do, right? And so I think this is where, all of these things are important to consider when you're deciding, what type of beekeeping you want to have, like incorporate in your operation.
Mhm. So if I throw a curveball at you, so you have a country home now and you've decided to keep bees in your own backyard. Do you currently keep bees, by the way?
No, I don't.
Okay, so now you've decided to. With all that you know, let's start with, how would you set up the 4 colonies that you want to keep, and what would your just basic outline of your management practices be in the state of Pennsylvania with a tiny backyard apiary of 4 colonies? What line of bees would you choose, and what would be your treatment plan? If it,
Yeah, that's a great, like I think the first thing that I would do would be to um and this is very hard, like from a practical, like perspective, but I think it's an important consideration, right? Like one needs to think about whether your landscape has enough flowers to keep your 4 colonies plus all the other bees that are out there, right? So I think that's something that I call, beekeeping is stewardship for the environment, right? Like you need to be sure that you can keep your colonies, alive and give you honey, but also, be friendly with everything, else that is using flowers. So, let's assume you have, like a place with lots of floral resources. I would definitely go for queens locally, like produced, right? We also have some research indicating that local queens are better than things that come from other places, at least, like here. So that would be my other recommendation. And I would definitely stick to organic treatment. So, at least one treatment a year, right? Um, like late summer, that's generally a must. But doing monthly mite washes, and when your mites are above threshold, rotate organic treatments.
Now, what do you consider the threshold for treatment to be?
Usually it's like 2 mites per 100 colonies,
Hmm. Two
right?
mites per 100 colonies? Or
Sorry, per hundred mites, per 100 bees, not
that
colonies. I'm
would
sorry,
be a
two
good
mites per
apiary.
100.
Two mites in 100 colonies, I would take it.
I'm sorry.
That's okay.
No. Um,
And so for your preferred organic treatment, does that kind of match with Dr. Underwood that you would use Formic Pro, or
yeah.
what's your impression about oxalic acid, or do you know anything about the new application of extended release Viroxan, for example, that people are using the strips?
Yeah, I've heard, I have not, like used them myself, but I've heard mixed results. And so I think beekeepers need to be very careful because, and I don't know if it is driven by climate,
Yep.
like, or the type of bee that you use. I don't know exactly what it is, but it seems to work really well for some and not work at all for others. So yeah, I think, like we probably need more experiments to
Mhm.
inform, like our local beekeeping community about the whether those treatments are effective or not.
And you mentioned Purdue. Are you involved at all with the Purdue ankle biter genetic line?
Yeah, we were, we collaborated with Brock on actually a queen comparison project that, included some ankle biter survivor stock and some Pennsylvania survivor stock. And that's the study that, like for us was clear that our regional queens, broadly speaking, regional, right, like outperform compared to queens that we bought from California or Georgia.
Mhm. So now let's backtrack to the pests that you found, because we made reference to pest loading in these feral colonies that were in the trees or other cavities that just weren't managed by people. What kind of pests are we talking? Strictly the Varroa destructor mite, or are we talking about other pests as well?
Well, I wish we were able to count mites, right? But for the feral colonies that are really limited, like if like making observations without disrupting the colony is very difficult. So what we did was basically collect samples of workers, foragers coming in and out of the cavities, and then we use molecular techniques to quantify several viruses and immune genes as well. And so indirectly, what I can tell you is that the in general, the feral colonies had higher levels of deformed wing virus, which indirectly tells you that probably mite levels were also, like higher in feral colonies, as you would expect.
Mhm.
Um, so yeah, that's how we figure out that, like those colonies had a higher pathogen burden.
Mhm. And this is where I think beekeepers and those that manage or don't manage honeybees kind of come under fire with those who are trying to protect the native populations of bees, being that the deformed wing virus is not species specific, so it can move from our honeybees to some of the native bee populations. Did you do any kind of studies, or do you know of any studies in the state of Pennsylvania regarding how DWV may be spreading and what
Hmm.
natural bees are being impacted.
Well, yes, we have looked at that. And like I don't want to downplay the importance of controlling the deforming virus because as we all experienced during COVID, right? Like we don't know when a new, like variant of a virus is going to become pathogenic and can, like end up being a pandemic and, like killing a lot of, like individuals. So, it is important to control the forming virus because of the potential for, like that spread. But in my research, what we have observed is that the forming virus is very prevalent. And what that means is that when we look at other bees, we find molecules of the forming virus in other bees. But when we looked at the infection levels, right, like whether the bee is really sick and the virus is replicating in other bees, we find like weaker evidence of that. Right. And so I think that that's something that is sometimes is not really well explained in the literature. There are a lot of inferences about risks about these, pathogen spillover and, like problem with diseases from honeybees to other bees. Mm-hmm. I think there is a potential risk, right, of pathogen spillover. But for the most part, right, like one of the big differences is that there are no other bees that can that have varroa mites, like attacking them or using them as hosts. Mm-hmm. And that is the big problem with deforming virus in honeybees, right? That we have the mites helping the virus replicate in honeybees. The other bees are getting, like sometimes infected through kind of like oral fecal route, which means that yes, like sometimes the virus can find bees and, like they can replicate, but the infection levels never get like what we see in honeybees, right? So I don't know if that is a convoluted answer to your question.
But... Well, it leads me to a follow-on question. So when we look at those virus loads that are formed wing virus on other bees, how are we sampling that? Are we getting that from a bee that's out on flowers? Are we getting into their nest material? And what about Vespidae? What about the wasp species and things that also may have it?
Yeah, so in general, the bumblebees have been much better, studied, and I think because bumblebees and honeybees are closer evolution, like they share a more recent common ancestor than, like with other bees. There is evidence that the four-wing virus can, like use bumblebees as hosts and replicate and have, like very, like negative effects on bumblebees. Mm-hmm. But, for other bees, some studies have actually done what we did, which was assessing the levels of these viruses in non-honeybees and non-bumblebees. Mm-hmm. But a lot of the literature is only giving information about the presence or absence of the virus. Okay. Which again, given the number of, like molecules of these viruses that are, are out there because of the mites, it doesn't surprise me that we're finding it everywhere, even in wasps. So regarding your question of wasps, I don't remember from the literature seeing studies quantifying viral levels in wasps. I know they have been detected in wasps, mm-hmm. but I don't know if there is strong evidence that they can really get infected with the virus.
Because I was taking apart a wasp nest, as many people do, the yellow jackets, and I found deformed wing virus on some of the specimens that were in the nest.
Oh, wow.
I did video it, by the way. If you want that, I'd be happy to shoot it your way if anybody's interested.
You should have collected samples and send them to me next time.
Now that I know, but Yeah, I was very curious because it was clearly not just that it had engaged and been chewed by another wasp or something. It was a lack of development of the wings. So it was very interesting to me. And that was a few years ago, but I record, photographically and with video, everything. So, do you actually go out in the field very much, or are you stuck at the
Mhm.
university? What are you doing?
Um, I am trying to get more to the field, and usually my field work is based actually in Colombia, where I run an undergraduate and it's a funded project where I take students from the US to Colombia to do research for 8 weeks. So that's usually the only time when I'm, like dedicated to doing research with students,
What
unfortunately.
kind of bees are you working with in Colombia? Still Apis mellifera? Or are you looking at those stingless bees and things down there? What are we doing?
Yeah, so it's a lot of wild bee research. Like working with honeybees in Colombia is not pleasant, let me tell you that, because they're super defensive. So, it requires a lot of, that's something that I don't think, like a student who has not experience, like with bee research would be. Yeah, I think it would be difficult to get students excited about bee research when you have, thousands of bees trying to sting you
Right.
anytime you open a colony.
Can you give us a story about when you encountered a highly defensive nest and what happened, how you managed that, how were you dressed, what happened to the, students that might have been with you? What should people know if they encounter? Because this actually in some parts of the United States is on the rise. So
Yes.
people are engaging with uh, honeybees that are behaving disproportionately aggressive. Like we used
Yeah.
to say just defensive, but I don't know. This is bridging into aggressive territory. Can you share a little bit about?
Yeah. I mean, so again, I have not really done, research with colonies, with honeybee colonies in Colombia, but I did actually my first ever, course about beekeeping was in Colombia with, Africanized bees. And you need to wear a, like full body bee suit, and you need to tape, your wrists and your ankles. And like I think the experience as a novice in, like the world of honeybees was, my adrenaline was, through the roof the entire time that, I was manipulating colonies, because, like, and you see the stingers in your veil, like it's
Breaking
yeah,
through,
it is
trying to get to you.
overwhelming.
Yeah,
Yeah.
yeah. And could you smell bananas? Did you smell that banana smell?
Yes. Yeah.
That is very interesting when you get them angry enough that there's enough of that in the air that really does smell like bananas.
Yes.
But it has nothing to do with actual banana oil, right? It just happens to be because based on that, people have said, don't eat bananas around bees, you'll get a response from them.
Yeah.
So how do they calm bees when because clearly they're trying to manage these really hot colonies? What is their best kind of practice for keeping them calm?
I mean, it is less management intensive, right? Like beekeepers don't, um, it's minimal, right? Like you go, you extract honey, you do, like you check your colonies every now and then, but you don't do a every 2 week mite assessment, like or anything like that, and they have to be like pretty separated, like spatially,
Mhm.
right? It is a very different world to work with highly defensive bees.
And so I realize this isn't a focus of your study, but I'm curious, just based on the feedback that I hear from people that are thinking, well, those bees don't have any mites and those bees don't have any pathogens. And so it's a trade-off. You get these really hostile bees that will kill all your livestock, but they are healthier. Is that, is there any truth to the Africanized genetics being more resistant to varroa mites and the pathogens that they carry?
Well, so okay, so here's the thing, right? Resistance is a word that is commonly used, but is often misused, right?
Okay.
Because resistance implies that the colonies or the host can reduce the level of a parasite, right? And so in my experience, and what I hear from beekeepers is Varroa is there, right? And it can be a problem, but often, like beekeepers are not treating as frequently as beekeepers need to treat here, right? And so I honestly think that there is a need to like have more research understanding mechanisms for like why that is the case. I guess I can tell you from the shared experiences with beekeepers in Colombia that varroa mites are less of a problem for beekeepers there than here. So whether that means that they are more resistant or not, I mean, they're there, right? It's not like varroa gets into an Africanized colony and doesn't reproduce. No, they're there. They get infected. Some get really high levels, but yeah, it is less of a problem for beekeepers in that region.
Now, are their colonies kept smaller? They have behaviors that are, some of the European bees don't. For example, supersedure, no, usurpation
Yeah,
is something that the Africanized genetics are prone to do. Do they stay smaller because they swarm more
I
frequently?
I think so. Like thinking of like apiaries I've been to. I don't think I have ever seen the size of colonies that we can reach here in
The
Pennsylvania.
big
I
stacks that
have a
people like to have.
Yeah, I have a photo of like Charlie Warsack. I don't know if you know him.
Oh, yeah, Charlie's in our
Well,
club. Yep.
yeah, so Charlie had, like for one of our experiments doing the queen comparison, he had like, I don't know, eight, like I think something like eight, boxes piled up in one of his colonies, produced an incredible amount of honey that year. So yeah, I don't think, like bees in South America could get that big.
No, I don't think I would want a colony that size personally. But now, so back to if you had your own backyard collection of bees, what style of hive would you prefer to keep based on what you've seen and know?
Hmm.
Like, would you do a vertical Langstroth? Would you have horizontal Langstroth? Do you maybe think about Lance hives or top bar hives, or what would be your preferred
Yeah, I think what I know best is, the vertical Langstroth.
standard Langstroth? Hmm.
Yeah, we are just, we just completed a an experiment, and I don't want to like say the wrong thing because I have not run stats on the data, but we compared honey production in single versus doubles. And what do you
Single
guess what
brood
we
and double
found?
brood, right?
Yes,
Okay.
yes. Any guesses?
For
Which
me,
colonies produce more honey?
the single brood.
You got it. Yeah, they seem to produce more honey.
Yeah, but
That
I'm cheating because Steve Rapaski. Um,
Well, Steve Rapaski was the instigator of this experiment because
there
we
you
were
go.
very skeptical of, like we need an experiment, like showing this. But I think, Steve doesn't know this, but I think he will be happy to see our results.
Okay. He does it out of pure convenience because he just wants all his brood in one deep box,
Yeah.
and then he's got a queen excluder and everything's above that. And he's south of us here, so I'm in a
Yes.
tougher climate. I have one colony that has two, it's a two colony system, two queens, central,
Mhm.
shared super. And I regret doing it because now I have to do work and take honey off when I don't want to. But those are highly productive. But the single brood, so you're saying, when will that study be published?
Um, we have the data, but I
I need to know.
cannot
I need to know.
predict, like there are so many things that, for example, this queen comparison study, we have not published it. And we have
Okay.
so much valuable information for beekeepers. So yeah, who knows? But hopefully soon.
Okay, so for those who are listening and watching, when these studies publish, you will find that link down in the video description, and then you can follow up and do a deeper dive. So that'll be cool.
Whenever that
So,
happens.
so what are you focused on now? Is that your current study? Is the brood box management size? What
Well,
else is going on?
yeah, so right now the size of my honeybee research is, like shrinking, and other aspects of my research are, like going in the opposite direction. But the main honeybee project is around organic beekeeping. So basically we have kind of like the phase two of this project where project one was all about organic beekeeping management and whether we could do that successfully and whether the profitability of something like that would be like something attractive for beekeepers. And we show that you can do it and that it is highly profitable. Then the next question is, can you actually get a certification for your products if you're using organic beekeeping management in your colony, right? And that is mainly driven by existing regulations, like coming for the organic board is like standards. And they, like one of the requirements is this, like huge area that needs to be pesticide free for your colonies to be, any product of your colony to be certified organic. So, we're focusing on studying foraging, and because like one of the things that we would like to demonstrate to policymakers in the world of organics is that the use of maximum foraging distance for honeybees is probably not really relevant for like the production of organic honeybee products, right? Like, so what I mean by that is that the fact that honeybees can fly very, very far away from the colony does not mean that most foragers are doing that, right? And we're collaborating with Maggie Cuvion from Virginia Tech, who has done beautiful studies on honeybee foraging behavior. And all of her studies show that most bees are really foraging about 800 meters outside of the colony, right? So it's not 10 kilometers like, like what these people are basing on, like of the policy. And so, yeah, so we're, like studying foraging behavior. And one of the things that we wanted to study was whether smaller colonies, because they would have, like lower energetic demands because they have, like smaller brood, would actually forage, closer to the colony or not. I don't have, an answer for you about that yet, but that was the premise of, like the study comparing single versus doubles. But yeah, we are, like the results of this study are very interesting, and I don't want to spoil it, but I think we have some data suggesting that it is probably feasible to establish organic beekeeping operations in continental US. And we just need, like support from policymakers and actually from beekeepers, because one of the things that we need to do, and we haven't really been very good at it yet, is we need support from stakeholders to demonstrate to the organic board that there's enough interest in organic beekeeping for them to even pay attention to any of this research. So
And just for some of the more beginner, listeners, they're wondering what we're talking about. But the point of getting an organic certified honey, like we don't see that on the shelf, and the reason being what you just described, they speculate that, well, they can go 2.5 miles in every direction, so that means every floral source they would encounter within that range would have to be certified organic as well, otherwise you can't get it. So then we find out, well, there's no place like that where we're not in range somewhere where they're engaging with something not organic. So you're saying this may change, that the regulations or the parameters as far as how we prove that. Do you think that's also going to improve based on the new technology in honey testing, that you would be able to know if they were visiting plants that were not organic?
what
Because
do
now they
you
have
mean?
these nuclear test systems where they can find components in the honey with greater accuracy than they ever have in the past. So I
Well,
wonder if that actually is a quicker way to certify someone's honey. If they're a sideliner or 500 or more colonies, um, then that kind of testing would get you that label through our.
well, this is something that I've learned with the very few interactions with policymakers in the world of organic agriculture. The emphasis is on the process and not on the product.
Okay.
So you do need to follow standards that they define. And sometimes they test products, but they cannot be testing everything. So this is
Right.
why the emphasis is on the process and not the product. So we do need to do need to convince them that, like the data and the science shows that you get comparable products here in Pennsylvania or New York State under certain parameters compared to, like things that we are importing and have the certified organic label.
Hmm. So you mentioned that your studies and focus on honeybees is shrinking and you have other areas of interest that are expanding. What are you moving into? What are you looking at?
Um, it's a little bit all over the place, but um, we have, like there are like three major, areas in addition to organic beekeeping in my lab at this time. One is we have been running a monitoring project for wild bees across the state. And basically, we have been collaborating with community scientists across the state to characterize bee biodiversity, across space and time. So now we have a much better understanding of who are the wild bees of Pennsylvania and, where they're found. Actually, I have a plug. We just published this new book, which
Oh,
you
yeah, I was just
can,
reaching for mine to have a bunch of those here.
yeah. So well, like this is a major, like a lot of effort goes into that project for a lot of reasons. We're collecting a lot of interesting data, and we are about to seek funding to basically launch this project at a national scale. So yeah, that is taking a lot of like my effort and attention at the moment. Um, we also do a lot of work with the squash bees, as you mentioned at the beginning,
Mhm.
because those bees are extremely interested. We are actually demonstrating that the bees have adapted to the recent changes in the landscape as agriculture has expanded and intensified.
Mhm.
So it's kind of a very interesting model system to demonstrate that bees can adapt to ag, and that not for every species agriculture is necessarily bad, and like we can have like both things coexisting.
Mhm.
um, and we are doing a lot of work with pathogens as well. So that's the third other theme. We're trying to do all the boring work that people don't want to do, trying to understand what these pathogens are really doing in other bees that are not honeybees. And so that's the other major research theme.
So then how are you selecting your citizen scientists that are going to participate in this kind of information or sampling?
We're very biased
Okay.
because like I feel like I've done, I mean, and we've talked about at least two other projects where we've had participation from the public.
Mhm.
But working with the beekeepers is great because beekeepers are invested in like the science that we're or the questions that we're trying to answer.
Mhm.
In a similar way, for all of these projects with wild bees, we have been working with master gardeners who
Master
like
gardeners.
are
Okay.
dedicated volunteers. They spend a lot of time working with us on these projects. So we have been able to demonstrate that you can work with community scientists and not compromise the quality of the data, which is always the challenge with community science. And some people are very skeptical of it for that reason.
Mhm. Okay. All right. Well, I'm not going to keep you a lot longer, but I would like you to share one thing about yourself that most people will not know. some hobby, some skill, some talent, something that you do or know about that people normally would not assign to you?
Hmm. I don't know. I guess I speak three languages. I play the piano, and I recently became a weightlifter.
You're a weightlifter? I want to focus
That's a
on
new
that.
thing.
So are you, all right, why are you lifting weights? Is this to put off osteoporosis? What are you doing? Are you a power lifter? What's going on?
Yeah, so I started lifting weights about a year ago, and I'm completely, like hooked with that. And why I'm doing it, I guess I've read science that, says that it is important to build muscle to have not only to not live longer, but live better.
Right.
So that's my motto for this.
Quality of life.
Yes.
Absolutely. But you don't want to give us your combined lift score or anything like that. You don't want to
No, I am an amateur, but like I'm very dedicated to it.
Okay, well, I really want to thank you for your time today and for sharing all that's going on and give us some insights to what your world is as far as research goes and what we're finding out. And I hope that people will come back and look in the comment section and will link studies that you have already published as well as when the others finally get in the public arena here that we'll be able to learn much more. So thanks so much for your time today.
Thank you, Fred.
Yep, absolutely.